Hey, Kent.
Aaron, hi, how are you doing?
Good to see you.
And you as well. So, Okinawa in June, is that what you said?
Yeah, I'll,
Okay, you're busy.
I'll sorry, no, I'll be back. I have a flight booked. How do I say this? I have a flight booked June 2nd to come back to San Francisco.
Oh, what's the occasion?
It's summer.
How long are you going to be here?
Um, yeah, I'm still working out all the details.
Will you be visitable?
Absolutely.
Okay, great.
And,
Go ahead.
and I've also been trying to get a visa to go to Okinawa in June for a neuroscience meeting.
Oh, you really get around.
Yeah.
Um, okay, well, do remind me if you would. Let me see, I'll write that down. June June 2nd, you will reappear here.
Yeah, and then give me some time to
Yeah, of course.
decompress.
Although, sometime in there, we're going to Japan ourselves. It's been 3 years since we saw Mama.
Great.
Mm.
Hi, Rui. Good to see you.
Okay, Erin, bye-bye.
Okay.
Hi, Aaron.
Hey, Rui. Good to see you.
Good to see you.
Do you want to test out your slides?
Yes. Can I try?
Yes, please. Yeah. Go ahead. And
Can I
then
see
can you do the yeah,
the full screen?
yeah. Yeah, that looks good.
Okay.
How's everything at Yale?
It's good. It's good so far. Yeah.
Great. 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 Thanks for giving a talk. Looking forward to hearing the latest. Hi, Manther. Good to see you.
How are you doing?
Good, good. How's everything going?
It's going. It's going well.
Cool. Nice. Do you want to test out your slides?
Yeah, I can do that. You guys see them?
Yeah, looks great. So we've become really interested in the VATPases. It'd be cool to chat about it. Yeah, that looks good.
Okay.
And then you could stop sharing whenever you're ready. I just have like an introductory slide, and then you'll be the first one up.
Trying to find
How's
this.
Stanford? Oh, here we go. Yeah. How's Stanford doing?
It's doing well. It's been like colder these days for some reason.
Yeah.
Oh, I found the stop sharing here.
Yeah, I'll be back in June.
That sounds great. How long you've been there?
It seems like forever, but about a year.
That's great.
Yeah. Right now, so I'm living in Shanghai and the whole city is on lockdown, so that's been a drag.
Are you with the family over there or on your own?
Family.
Nice.
Hi, it's Long.
Hi,
Good to see you.
let's see. Yeah.
How's everything going there? It's Long.
Yeah, not bad, not bad. Hope we're still dreaming about to come back and this month or next month to work.
I think Pudong is cheating on the nucleic acid testing because
Yeah.
we had a nucleic acid test yesterday,
Uh-huh.
and then they said we're going to do another one today, but you can only take it if your one from yesterday is negative, and you take an antigen test and it's negative, and you haven't been out for medical treatment in 5 days. So it's sort of like forcing it to be
Yeah.
negative. So of course the numbers, and then because the numbers will come down if you do that.
This is pretty chaos. That's
So I support that if it'll let us get out earlier, but it seems a little bit of a cheat.
it's, it seems today's number going down for like 2000s or 2700. Hopefully, it's still if that could get trendy, still could come back.
Yeah.
Don't know.
Yeah. But then they just, yeah. So they said like, because we took a nucleic acid test yesterday, and then they contacted us and say you have to take an antigen test tonight, and if your tests are both negative, then you can take a test tomorrow. But if not, so it's sort of like tricking the system into getting all negative.
Yeah, sometimes it's pointless to get the antigen test at all time. Twice a day first.
You have to take it twice a day.
Yeah, it's last week.
Wow.
What's the point of that?
Yeah. We should have started a antigen test company.
Yeah, but still people complaining some false negative, false positives. Sometimes not very reliable.
Yeah.
But there are some in Europe, US, right? I heard similar antigen tests you can do it at home.
Yeah.
My friend in Germany can do that, and really,
I'd be willing to take a test every day or twice a day if I could leave my apartment.
yeah, definitely.
I'd trade that. Yeah. But maybe in a few months we'll get out.
Yeah.
So where are you, Aaron?
Oh, hey, Judith.
China?
I'm
Hi.
in China. I'm in China. Yeah.
And you
Yeah,
can't
it's pretty
leave your
crazy.
apartment?
Nowadays I can leave my apartment to walk around the compound. It's like a neighborhood, but other people don't have it as good. But you can't go to the store or anything like that.
So how do you get food?
This is a good question. This is like the main focus. Like I stress out about grants and papers and airline status, but my main stress is how to find food. I was trading like cigarettes for fruit and like grapes for this and some
Cigarettes.
neighborhood. Yeah, cigarettes are good. Beer is good, and yeah. And then some people can organize group purchases. So you can say, oh, we want to buy like, I don't know, 100 packs of this or that.
Cigarettes.
Yeah. But hopefully this will be over soon.
Yeah.
But it's a good experience, I think, experiencing the whole range of Shanghai for the whole year.
Yeah, it's been a long time memory. Maybe
Yes.
34 years ago, we don't have any coffee, don't have any some new fancy
So
food.
like, because I remember like when I was little, my grandparents would be like always saving things because they were poor, they never had stuff. And now because we're running out of like plastic wrap and aluminum foil, I'm like saving like the aluminum foil like to reuse, and like coffee I'm like using a little bit less because I'm running out of that.
Yeah.
So
Right,
maybe
right.
I'll keep remembering that. Yeah.
I get started?
Okay. Yeah. Okay. Enough of that miserable news. So it's great to see everyone here. Even though Shanghai is locked down, we'll still get together and talk about great science. Looks like Beijing is going to be locking down soon. So Zilong and I will be here with you for as long as it takes to talk about science, forget about our hunger and things like that. We have awesome speakers coming up, and before those speakers, I want to just advertise that next week we have more great speakers. Baptiste Lacoste from University of Ottawa, talking about blood-brain barrier and astrocytes, and Zhong Ye from ION, talking about working memory. So please tune in for those awesome talks. Oh, great news! A long-term friend of NeuroZoom, Professor Ishi Jin, elected to the National Academy of Sciences. So that's great. So if you give a talk in NeuroZoom and attend, good things could happen to you too. So with that, I'd like to introduce our first speaker, my good colleague at Stanford, Manther Abu Ramalia. He's an assistant professor of chemical engineering, and he has a courtesy appointment in genetics. I don't know exactly what a courtesy appointment is, but for all intents and purposes, he's also a member of the genetics department. Manther received his undergraduate degree in Jordan University of Science and Technology, but not just a degree. He got the top rank in his class, and then he did a master's and PhD at Hebrew University of Jerusalem, where he explored the role of transcription factors OCT3/4 in regulating Wnt signaling and how it controls stem cell fate. He then did a postdoctoral fellowship at the best institute in the world, the Whitehead Institute, with David Sabatini. Here, I lost track of how many papers in Cell Science and Nature. I think it's like 9 papers during his postdoc, including a major first author paper in Science, in which Manther invented a technology that lets him take organelles like the lysosome and use it in a very rapid way and profile the metabolites that are in there. And you could do this under different conditions. And he used it to discover how unexpectedly the lysosome regulates the efflux of amino acids differently under different stresses. And the lysosome is a major focus now of neurodegenerative disease, and Manther has the tools to really solve important problems. So it's wonderful having him as a colleague, and looking forward to hearing his latest. Go ahead, Manther.
Thank you very much, Aaron, for a really kind introduction. So as Aaron mentioned, we've been using some what we call lysosomal immunopurification tools to study early onset neurodegeneration, and this is the story that I will be sharing with you today. So my lab in general is really interested in studying subcellular metabolism, and as you might know, most of the important biochemical reactions in the cell are happening within compartments, and the reason for that is that the compartments would provide the optimal conditions for these reactions to proceed. And there is no one compartment that is a good fit for this description as the lysosome. And the reason for that is the very acidic environment of the lysosome and the content of this organelle, which would allow the degradation of almost every single type of biomolecules, because it does have these 60 different hydrolases, and they operate under pH optimum of 5 or 4.5, which is the pH of the lysosomal compartment. And my lab, since started like 2.5 years ago in Stanford, is really interested in trying to understand how the lysosome contribute to the overall metabolic network in the cell. So we've learned a lot over the last few decades about how the lysosome degrades the different macromolecules, but we know very little about how these macromolecules, how the output of this degradation process is being used in the cell, and whether this process of the efflux of different metabolites is being regulated, and whether this regulation is somehow being coordinated through signaling pathways that would allow the lysosome to fulfill the metabolic demands of the cell. For example, if you degrade proteins, whether the amino acids will be stored in the lysosome or they will be in equilibrium all the time with the cytoplasm, or this process is regulated in some way or another. And this major interest came from this fact here, that any problem with the lysosomal function, a state that we call it lysosomal dysfunction, and this could be trafficking macromolecules to the lysosome for degradation, the process of degrading these macromolecules into their monomer content, or the efflux of these monomers to the cytosol to be utilized, is called lysosomal dysfunction. So these problems, once happen, they affect the organismal homeostasis and lead to human diseases, which I like to categorize into two groups. In one of them, we talk about the monogenic diseases. In this case, we have mutations, biallelic mutations in the lysosomal genes inherited from parents. The kids usually will have a lysosomal storage disease. And we have around 55 to 60 of these lysosomal storage diseases. And in many cases, almost 60 to 70%, there is some sort of neuropathia involved or manifest in these kids. While these are really ultra-rare diseases in most cases, it turns out that the lysosomal dysfunction is also a hallmark of several age-associated neurodegenerative diseases. And we see this, we observe this in Alzheimer's disease. So at the histological level, we clearly see that the endolysosomal pathway is an early implicated pathway in the disease, especially from post-mortem slides that people have been looking at. In Parkinson's disease and from frontotemporal dementia, we have even more genetic element of the lysosomal pathway being involved, as seen here. So as an example, I'm just looking here at Parkinson's disease. When you look at the genes that either predispose for Parkinson's disease or genes that are found in familial Parkinsonism, or even at genes that seem to be mutated when exon sequencing has been undertaken on idiopathic patients of Parkinson's disease, you clearly see enrichment of the endolysosomal pathway genes. And as you see here, like, for example, we have either regulators of the endocytic pathway or the autophagy pathway. We also see clear core lysosomal protein genes being mutated as well. What's interesting, many of these, or even most of them, are genes that are mutated in these monogenic lysosomal storage diseases. But in that case, we have biallelic mutations. Both alleles are defective. In these cases where we have predisposition for Parkinson's disease, you would have one allele being mutated and the second one is functional. So it seems like over time, the other allele, which is functional, is not enough to perform the lysosomal function that should be performed. At least this is the hypothesis in the field. So with this in mind, when I started my lab, we thought maybe we can use these monogenic, well-defined diseases in terms of genetics. We know exactly what gene is being involved or being mutated, to try to understand what exactly this lysosomal dysfunction at the biochemical level. And maybe through this understanding, we can shed some light on these very common diseases, including the age-associated one listed here. So the exact question that we are trying to address is what exactly the lysosomal dysfunction at the molecular level. And to do so, we realized that we need to be able to study the lysosomal content. So if you think about all these lysosomal enzymes, transporters, and other machineries in the lysosome, the major functional output is the monomers being generated from the degradation process, and probably some other processes that yet to be discovered. So to do so, during my postdoc, we developed this method that allows us to pull down lysosome from cells back then, and now we can do it in tissues. And the principle is really very simple, and it was discussed earlier in the literature as well, which is based on the immunopurification of organelles. So as you know, in biochemistry, you can pull down proteins and look at their interactome. You can do the same with organelles by simply expressing a tag or a lysosomal, in this case, lysosomal membrane protein TMEM192 fused to epitope, which we chose to be the HA epitope. Then you express this in any cell type you are interested in studying by simple homogenization using very simple buffers. You can release the organelles. You come with anti-HA magnetic beads, which would allow you to bind these lysosomes. And then using the magnet, you can take the lysosomes to the side, wash away all the other organelles and cellular content, end up with these intact and pure lysosomes that you can take them for any type of analysis. So we were able to do metabolite profiling, proteomics, even RNA-related stuff with these purified lysosomes. So when we developed this method, the major question that you would have in mind, is this method quantitative enough to see changes in the lysosomal metabolite profile, since it was really the first time looking at the lysosomal metabolome at that time. And to do this, we utilized a very simple trick in the lysosome biology field. As I mentioned, the lysosomal lumen is very acidic. And this acidity is maintained by this VAT-PASE machinery that would pump the protons into the lysosomal lumen, creating a proton gradient across the membrane. And this proton gradient acts as a driving force for many transporters to transport the lysosomal metabolites to the outside of the lysosome or to the cytosol. You can easily inhibit in a selective manner the VAT-PASE using simple drugs like bufalomycin and concanomycin in this case. So we took these drugs, we throw them on the cell, and then did metabolite profiling. And when you look at the changes at the cellular level, you barely see any change, almost no change at all. But when you take lysosomes using the lyso-IP method, from these same exact cells, you see this very dramatic change in their lysosomal metabolite content. And this was really very satisfying result because it says two things. The first one, you really need, if you're working with organelle that is only 1 to 3% of the total cell volume, you really need some sort of subcellular metabolomics to be able to see changes. And I don't imagine that there are really stronger hits for the lysosome than the VAT-PASE machinery being hit. So it's really important to keep this in mind, and you will see this also as a pattern in the coming study that I will present, because you don't really see any change at the whole cell level. You see this massive change at the lysosomal level. And the second thing, of course, which is why we did this experiment, is to show that the lyso-IP metabolomics can detect these changes in the lysosomes. So having this tool in mind and having also this approach in our hands, we decided to go after lysosomal storage diseases, as I mentioned earlier, using very simple models, really artificial models in this case, where we can take any cell type. In this case, we are using KEC293Ts, where we use CRISPR to generate mutants or knockouts of the different lysosomal storage disease genes. And people have done this a lot, looking at the whole cell. Now we can look at the lysosome as well. And this work was mostly done by me and then a postdoc in my lab, Noflaktoum, where we decided to focus on this group of diseases called neuronal steroid lipofuscinosis, caused by mutations in almost 14 different genes. All of them encode either core lysosomal proteins or proteins that regulate the lysosomal function. And as you might imagine, this is neurodegenerative disease, and that's why it's called neuronal. Steroid are these aggregates that accumulate in the lysosomes and then spread in the cell. And lipofuscin coming from the word lipid and autofluorescent. So these are autofluorescent aggregates in the lysosomes. And this is the same dye that people look at when they look at aged tissues. So when you look at aged tissues, you see a very linear correlation between the age and the accumulation of lipofuscin, even during normal aging. So it seems like, or people think about these diseases as accelerated aging type of disease. So we focused on CLN3, and I will tell you why, but it's causing the most common form of these neuronal steroid lipofuscinosis called Batten disease. It's very severe and very nasty disease. By the age, at the beginning, there is no effect. The kids are normal, but then when they hit like 3 to 4 years old, they start to show some signs of a disease. And this usually starts by vision failure, followed by very progressive mental decline, seizures, and loss of motor skills, leading to early death by the age of 20. And of course, this is associated with neuronal cell death, as you see in this illustration. So the gene encodes this lysosomal membrane protein. Here it's drawn as 6 transmembrane protein, but it's actually 11 based on the AlphaFold predictions that we've done recently. So it turns out that this is a lysosomal membrane protein. We know this fact for almost 30 years, more than 30 years now, but we don't know its function. And similar to others, we propose that it might be a transporter of some sort. It could be an import-exporter. In this case, you would imagine that substrates will accumulate in the lysosome if you knock it out. It could be an importer importing some sort of substrates into the lysosome. And in this case, you will see their depletion once you knock out the CLN3. So it's really a very simple model that we can easily test using the lyso-IP method, because you can knock out CLN3 in cells, pull down lysosomes, and then do this metabolomics or metabolite profiling analysis. So we did this. We took the HEC293 T cells. We knocked out CLN3 and then pulled down lysosomes. And these are 2 different clones here compared to wild type. And we actually did not see much of a change, at least not a reproducible change between the 2 different clones that we have here, clone number 1 and clone number 2. It turns out that when you, and of course, in this case, we are doing targeted metabolite profiling. As you might imagine, when you set up a platform for metabolomics, usually you look at metabolites that everyone is studying, right? Because these are the ones that you want to detect in a targeted manner. And that's where we started to think, maybe we are not seeing changes because we are looking at metabolites that everyone is studying. If these were the ones to be different in CLN3, this story would have been already discovered, exactly what CLN3 is doing. And that's where we started to develop these untargeted metabolomics approaches for the purified lysosomes, and taking the same exact cells, putting down lysosomes, and then put them on the mass spec, or liquid chromatography mass spectrometry in this case, doing untargeted metabolomics and looking at ions that are different between the wild type and the knockout lysosomes. We found massive accumulation of these metabolites here, a group of metabolites. And a few months later, we were able to use analytical chemistry tools and define exactly what kind of metabolites accumulating in these lysosomes. It turns out that these are catabolites from phospholipid degradation. So as you all know, the major component of cellular membranes are these phospholipids, where you have the glycerol backbone, phosphohead group that defines the identity of these phospholipids, and then 2 acyl groups attached to them. So if you cleave the 2 acyl groups during hydrolysis, you release these glycerophosphodiester's. And these are really the metabolites accumulating in these lysosomes from CLN3. And we find that all of them are accumulating as a family of metabolites. So these ones here are these glycerophosphodiester's, which I will call them GPT going, or GPTs going forward. So that was really exciting for multiple reasons. First, we don't know of any mammalian transporter of GPTs in the mammalian system. The second thing, the person who discovered the lysosome, Christian de Duve, back in the 1960s, already showed that the lysosome has a phospholipase activity. I'll just go back here to show you what he found. So de Duve discovered that the lysosome has a phospholipase activity that is predominantly the one cleaving these 2 acyl groups, releasing the GPTs. And he said back then that these are the end products in the lysosomal degradation of lipids, and then they need to find their way out of the lysosome. And that's why we were really excited about this fundamental finding. So just to show you the power of the lyso-IP here, and that this phenotype is really a lysosomal phenotype, when you look at the whole cell changes, you barely see any difference between the wild type and the knockout. So now we are just looking at these same dots in a targeted manner to validate that what we are claiming is true. You look at the lysosome, and then you look at these GPDs. This is one example of them. This massive accumulation is observed. You add back the CLN3, you completely rescue the phenotype. Of course, this holds true for all of them. So again, similar to that VATPase inhibition experiment that we've done, you barely see any change at the whole cell level. And you really need to dig into the lysosome to be able to see that metabolic pathway that is perturbed in these diseases. So as I mentioned earlier, this is completely artificial. We are using HEC293 T cells. They have nothing to do with the neurons and brain and whatever caused the disease in this case. And that's why we decided to build this animal model, which we call lysotag, that allow us to express the tag in any cell type we are interested in. So we have a lock-stop-lox cassette preceding this lysotag, the TMEM192 3XHA protein. And what we did for this specific experiment, we just expressed it in all cell types by mating it or crossing it with the CMV Cree, and then validated the fact that we can do lyso-IP from tissues because it's not trivial. And we started, of course, with the brain because this is the one we are interested in. And as you can see here, when you do a lysotag, whether you are using a CLN3 wild type or knockout lysotag mouse, you clearly see that we can enrich for the lysosomal compartment. And this compartment that we enrich for is very clean from all the other organelles or contaminants. As I just mentioned, we crossed the lysotag with the CLN3 knockout mouse. We improved our lyso-IP to work for metabolomics as well. And these are really quick experiments. And what we found that when you take the metabolites from the lysosomes derived from the brain, you see a very nice separation between the lysosomes here. This is heterozygous. So they do express CLN3 versus the knockout, but at the whole cell or whole brain level, you barely see any change. When you look what is different, you find these metabolites accumulating in the CLN3 knockout cell brains. And then you look at the nature of these metabolites. Again, these are the same GPDs that we found in cells, which is really a nice confirmation of the results. So what we've been thinking about so far is that loss of CLN3 leads to accumulation of these GPDs in the cell, but we wanted ways to look if this is a flux problem. Once you take CLN3 out of the lysosome, these GPDs have no way to get outside of the lysosome and not some other artifact. And this is where we utilized our expertise in isotope tracing. So what you can do, you can send lipids directly to the lysosome by conjugating them to the albumin, and then you let the lysosome cleave these lipids. And these lipids would be deuterated, which would allow us on the mass spec to see them as a different intensity than the natural lipids that exist in the cell. So in this case, we used phosphatidylglycerol, which has a deuterated head group. And what we are trying to trace is the accumulation of the GPD part of this phosphatidylglycerol after cleavage in the lysosome. And the experiment is really easy. You just put these on the cells, and then you can do lyso-IB and look at the deuterated form of GPG. And you clearly see that it's massively trapped inside the lysosome. It doesn't find its way out when you have a knockout cell in this case. Of course, you start to see the signal at the whole cell level because this is not part of the cellular milieu. And that's why if it accumulates in the lysosome, you also see it at the whole cell level. So our model so far would say that if you have these GPDs trapped in the lysosome, so it means that their nutrient content, including choline, glycerol, inositol, would be less contributing to the rest of the cell, at least the lipid biosynthesis in the cell. And to test this model, what we utilized in this case, the phosphatidylcholine. So similar experiment, but doing this in neurons this time. What we do, we conjugated this phosphatidylcholine to albumin. And again, the choline part of this lipid is deuterated. So we can trace the choline in the rest of the cell and trying to see if there is really decrease in CLN3 knockout versus wild type, or this, in this case, the heterozygous. And indeed, when you put these on the cells, they make it to the lysosome. The lysosome would cleave the GPD, so it's releasing the 2-SL group. GPC will make it out to the cytosol where we have these glycerophosphodiesterases. And you clearly see much lower levels of these D9 choline. Same for phosphocholine, which is the next in line in terms of molecular component of the choline pathway. But what's important is what's happening next. So if you have less of this deuterated choline, do we really see any effect on lipid biosynthesis? Because you would imagine that these will be incorporated in lipids that are being biosynthesized in these cells. And indeed, when you look at any choline-containing lipids, whether these are phosphatidylcholines or sphingomyelin, you always see much lower contribution of these deuterated cholines to these lipids, which means that the lysosome is supplying less. What was striking to me, to be honest, when I look at this data, the media itself does contain choline, but it's clear that the neurons are heavily dependent, at least in this setting that we have here, on lysosomal supply of choline, which is something that we are studying heavily in our lab. Of course, you might think, okay, maybe it's the lipid turnover in these cells is different. Maybe there is a problem in the biosynthesis of lipids and it has nothing to do with the supply through the lysosomal pathway. It's very easy to test for that. You just take the same neurons or the same cells and you trace with D9 choline, which will make it directly through the plasma membrane transporter into the cell. And in this case, there is no difference between the wild type and the knockout neurons, which is really very important control to have to say that it's the lysosomal supply of choline that is being perturbed. So far, the way we think about the model is that loss of CLN3 leads to accumulation of these GPTs. And the question, where is the toxicity coming from? Why we have neurodegeneration? One hypothesis is the lack of these nutrients important for the cell. And we have some data showing that indeed we have lack of these nutrients, as you saw from choline, but we still don't know if this is the toxic effect. The other thing that we've been thinking about recently is the fact that massive accumulation of GPDs might be inhibiting upstream enzymes through a simple product inhibition hypothesis here, leading to the accumulation of these lipids. Because you have phospholipids that will be deacylated. When you remove one acyl group, you end up with these lysophospholipids that then deacylated to make these GPTs. So we did lipidomics on these lysosomes as well from the brain, and we clearly see accumulation of several species of these lysophospholipids, which are known to be very toxic for the cell because they have this weird physical property that can disrupt the membranes, including the lysosomal membrane. So we are still investigating this specific phenotype, but what we think about now, it could be that accumulation of GPD itself is toxic for the lysosome, leading to some sort of lysosome-mediated cell death. It could be that upstream accumulation of these lipids, which is something that we are trying to investigate as well, or lack of nutrients coming out from the lysosome through CLN3. But to answer all these questions, we need to know what are these exact enzymes here, which turns out to be unknown in the mammalian system. And this is something that we are currently investigating in the lab. But at least the data that we have now is providing this model that we can now test and trying to understand the major cause of neurotoxicity in Batten disease. And just to leave you with this before finishing my talk, we collaborated with Forbes Porter lab at the NIH, which is leading this natural history study of Batten disease patients. And we looked at the CSF, and the metabolite that we can, or the GPT that we can see in the CSF is GPI, glycerophosphainositol. And if you look here, it's really clearly differentiating between Batten disease patients and all the other patients that they have from different disease groups, which is really nice because there is no biomarker for the Batten disease. And we hope that this would provide one that we are now following up and trying to see if it correlates with any progression or other disease phenotypes. And finally, we think that this pathway might be a converging pathway of other NCL genes. And this is something that we are now studying in the lab, trying to look at CLN7 and CLN11 and other NCL genes that are leading to neuronal steroid lipofuscinosis. With that, I would like to thank my lab, especially No Flaktoum, who really led this project, Wen Tao Dong, our metabolomics expert, and isotope tracing expert as well. All these studies for tracing won't be done without his expertise. And thank you very much for listening, and thanks, Aaron, for the invitation to speak here.
Okay, thanks so much, Manther. Awesome talk. This is the funnest part of being at Stanford, is amazing colleagues who are just making cutting edge discoveries. I'm always inspired. So awesome talk. We have time for questions. Kang, please.
Manther, great talk. So you said the GPI is also likely to be accumulating in the CLN3, right? So there are many proteins that are GPI linked, and then there are subcellular localizations require GPI. So I was wondering if you have looked at any of these proteins, ER localized proteins or mitochondrial localized proteins, and to see, or plasma membrane. Does it? I can imagine if there's major problem with GPI link targeting, protein targeting, and the neuron is probably in bad shape.
Yeah, just the short answer for that, this is a different GPI from the anchor that people always talk about. This is actually the glycerol with the phosphoinositol. So it's the degradation product of the PI. So yeah, I think that the abbreviation is a bit misleading. People start to think about GPI as the GPI anchor. Probably I should start to write it as the full name instead.
Hey, Manther, I have a question. So towards the end, you showed the heterozygous CLN3, and it didn't look like there was a major change. I'm just thinking about how heterozygous mutations in another one of these genes, CLN11, also known as progranulin, can cause frontotemporal dementia in the heterozygous situation, but NCL in the homozygous situation. Have you looked in cells with those mutations?
So we are studying GRN in the lab right now, and we have mouse models for homozygous and heterozygous, trying really to answer this exact question, whether there is a dose dependence. So we still don't know if there is a very specific phenotype for GRN. We didn't get to do the experiment similar to the CLN3 one, but this is a question that we are really trying to address for both GRN, and we are looking at GBA, which, as you might imagine, is also very interesting because people are thinking about GBA as heterozygous being a predisposing factor for Parkinson's. And I don't think there is a clear evidence that there is accumulation of the substrate of GBA yet. So hopefully in lysosome, you would see something like that.
Right. I think your tool is going to be an amazingly powerful way of phenotyping cells in animals with these mutations.
Thank you.
Mother, I have a quick question.
For
the Batten disease, did you show the first until 5 years the patient got to be blinded? Am I correct?
Yeah,
that's suggesting. Right, this is suggesting CLN3 plays some important role in the visual system. What kind of particular cell, or like would you kind of speculate what's going on here?
Yeah, there are actually two different hypotheses. One is more talking about the optic nerve being affected, and the other one is looking at the retinal epithelium. So there is more work now in the epithelium showing that they are also affected. So it's not really clear where the blindness is coming from, but both cells are affected by CLN3 loss. Oh. And seems like the lipofuscin is heavily accumulating in that epithelium as well.
Okay, thanks.
Manther, do you see these sorts of accumulations in mice or cells with LRRK2 mutations?
We don't see, no, they are really specific. So we've done LRRK2 and we don't see GPDs accumulating.
Wow, okay. Yeah, the rescue you showed is really stunning.
Manther, is it surprising that by just tagging a transmembrane protein that you can purify lysosomes so successfully? I'm just wondering, these proteins are synthesized in the ER, they have to go through the biosynthetic pathways. Does that mean that the protein itself is just very much enriched in the lysosomes, or did you have to do some other tricks? Like in your homogenization, and did you fractionate, sucrose gradient? Did you do any of those?
Yes.
No?
So the goal is really not to go through any of those. Otherwise, you would lose the metabolites in most cases. This is like a 10-minute kind of IB, although it's really misleading when you say 10 minutes because we do it per plate. So each plate is 10 minutes, and usually the students will be excited. It's a very quick experiment, but then they have an experiment of 20 plates. So it takes the whole day because you need to do it one by one to make sure that you don't lose these metabolites or let things sit on ice for a long time. So for the protein, we worked with LAMP1, for example, and other groups before us actually tried LAMP1, and it does purify lysosomes as well, but they are not as pure as TMM192. So if you look at the LAMP1 purified lysosomes, you get too much of Golgi. And I think it depends on how it's being trafficked through the trans-Golgi network. For some reason, TMM192, which we don't really know why, it's one of the most lysosomal specific. You never see it on the membrane. If you express LAMP1 or LAMP2, you see some leakage to the membrane under high expression conditions. But TMM192 is, I think it's kind of luck. We got really one that is very specific to the lysosome. The other question is like whether TMM192 is targeting all the lysosome in the same way, or there are subpopulations. We don't really know. So, but this is something that we always keep an eye on, and we always talk to many collaborators are using our tool, and I always hear back from them about different challenges and different types of analysis they do to really find out if there's any of that happening. But so far, it's been really great tool to compare to different genotypes and also study lysosome under different conditions.
Awesome, thank you.
Okay. Suzanne, do you have any questions or comments? No? Okay. Suzanne Pfeffer's world expert in endolysosome system. Okay, thanks so much, Manther. Really inspiring talk. And Zilong, you're up to introduce Ray.
Thank you, Manther. Great talks. So it's my great pleasure to introduce Ray Chang, who is now assistant professor at Yale. So Ray has graduated from Tsinghua University in China in 2005. And after that, he went to California to do the PhD with the Emily Lehman in the University of Southern California, working on ion channels and taste receptors. And after PhD, he went to Harvard, worked with Steve Liebler's, started to study the vagal system and discovered some subtype of vagal neurons regulating the breathing. It's a very fundamental metabolic properties. In 2018, Ray started his own lab in Yale, and where he continued to discover a much more interesting role in the vagal system. You can see there's today Ray will bring us his new discovery, his new Nature paper, the multidimensional coding architecture of the vagal interoceptive system. So welcome, Ray. Take it away.
Thanks very much. Can you hear? Can you see the slides?
Yeah. Take it away.
Thanks very much, Sloan, for this very nice introduction. It's really my honor to have this wonderful opportunity to present our work here in this great system. So my lab at Yale is interested in the interactions between the visceral organs and the nervous system, and how these interactions can change our physiology and behaviors. And today I'm going to share with you a story that we recently published describing how we think that diverse changes in the visceral organs are communicated to the brain by this very important interoceptive system, which is our favorite vagus nerve, in a very efficient and effective manner. So interoception is the ability to sense the changes inside our bodies. And this is critical for our survival, and we are really good at this. For example, we can detect every single heartbeat and provide a beat-to-beat regulation of the cardiovascular functions. We can monitor the inhaled, we constantly monitor the inhaled air contents And if there are there are irritants inhaled, we make defensive responses such as cough. There are extensive innovations, sensory innovations along the gastrointestinal tract, where they detect the ingested food contents and help to regulate digestive functions. And we can also sense these released chemicals, including hormones and inflammatory cues. And many of these interoceptive signals are sensed or communicated to the brain via this single nerve, which is the vagus nerve. So the vagus nerve is a tense cranial nerve. In addition to, it's very well known in this parasympathetic control organ functions. This nerve contains many sensory neurons that sense monitor the information from all over the body, including the lung, the heart, liver, kidney, stomach, intestine, and many other organs that's not listed here. And the sensory neurons, the cell-bodies, are located in a pair of ganglion called the nodose and jugular ganglion, right across the jugular foramen. And these neurons are called unipolar neurons, similar like dorsal ganglion neurons. So they, on one side, they project their long axons to the peripheral organs, and on the other side, they project into this quite specific region in the brainstem called the nucleus of solitary tract. So as I mentioned earlier, that these sensory neurons in the vagus nerve can detect a large variety of changes happening inside the body. So you can imagine that this is a very complicated and heterogeneous system. And not surprisingly, there's a lot of studies recently focusing on trying to understand the molecular and cellular mechanisms of how these neurons are sensing different changes from different organs and regulate the physiological functions in the respiratory, in the cardiovascular, in the digestive, metabolic, and even in the cognitive systems. So just give you a very brief idea of this is a work I did with when I was a postdoc in Steve Liberty's lab and trying to understand the vagal control of the respiratory functions. So you can see that here we are using optogenetics to specifically stimulate the sensory arm of the vagus nerve. And before the stimulation, you see the animal has a very normal breathing patterns. And once we use optogenetics to stimulate these sensory neurons, the animal stop breathing right away. And once after the stimulation is gone, the animal starts to breathe again. So this is just a very brief example of the function of these neurons in controlling different physiological functions. And despite a lot of recent studies about the system, still there's very little known about how the sensory neuron heterogeneity and diversity actually facilitates this interoceptive coding at the systems level. We get a hint from one of our studies a couple of years ago that in Steve's lab, we try to image these neuronal activities in vivo and looking at how they respond to different challenges in different organs. And now you can see that the red neurons represent the ones who respond to stomach stretch, blue ones represent the ones who respond to intestinal perfusion of chemicals, and the green one respond to lung inflation. You can see when they merge together, there are different neurons respond to different things, suggesting there are parallel streams in the sensory vagus nerve. That presenting the information to the brain. So here's a question that I'm always interested, which is how is this system organized really to present so many different signals to the brain in a very efficient and precise manner? So we decided to take a step back and really look at what signals we think that needs to be coded by these sensory neurons so the brain can very efficiently and discriminate these signals from different, that represents different changes. And all the work I'm presenting here, most of the work I'm presenting here is done by a very talented postdoc, Chang, in my lab. So we think there are at least 3 key different features that the brain needs to know about these interoceptive signals. So first is the visceral organ, like the signals from different organs mean different physiological changes. For example, stretching the blood vessels versus stretching the stomach means there's either change of blood pressure or there's probably ingesting food. So you need to do corresponding changes in physiological conditions. A second feature we feel like the signals from even the signals from the same organ, if they generated from different locations or different tissue compositions, we call the tissue layers, it also means different change of different things. And the third one is, of course, sensory modality, meaning what the signal is, whether it's a mechanical change or this is a chemical change. Or if it's a chemical change, what type of chemical change. We reason that a faithful coding of these features in the sensory neurons is probably necessary for accurate signal discrimination in the brain. In the following slides, I'm going to show you how we show that if and how these different features are coded within the vagosensory neurons. So first, we wonder, how does the visceral organ, how is the visceral organ coded in these neurons? And because there's visceral topic maps of visceral organs in many different systems, for example, in the DRGs or in the sympathetic chain. So we wonder if there's a similar topographic map within the ganglia of the vagus nerve. So for this purpose, centrally injected retrograde AAVs encoding different fluorescent reporters, in this case, the T tomato into the heart and the GFP into the stomach, so that after a week, these sensory neurons can be labeled from the project to different organs, can be retrogradely labeled, and we can visualize them. So you can see that largely there are different neurons innervating different visual organs, suggesting that the organs indeed coded within the system. However, when we look at their distributions within the ganglion, it looks like this is a salt and pepper pattern, meaning that these neurons are randomly distributed within the ganglion. So then we think about maybe if this is not a spatial organization, maybe this is a genetic, coded by genetics. So we essentially developed this a tool we call the projection-seq in collaboration with Li Zhang, which is in the Department of Neurology at Yale School of Medicine, who is an expert in single-cell omics. So the idea is really simple. So we inserted a barcode, we call the unique projection barcode, into the retrograde AAVs, so that and we can inject different the virus with different barcodes into different organs within the same animal. In this case, injected 7 organs, including pancreas, colon, duodenum, stomach, esophagus, heart, and lung, in the same animal. So that after a week of infection, he can collect the ganglion and dissociate them into individual neurons and then sequence them using 10x genomics. And because the neurons are labeled with the barcode, so in the following analysis, we can know which neuron projects to which organ based on the barcodes expressed. So here I'm showing you, this is a UMAP plot of around 15,000 sensory neurons, legal sensory neurons, color-coded by their target organs. So not only that, from this study, not only we can see that the neurons innervating different physiological systems are largely genetically segregated, you can find the genes that specifically expressed in the neurons innervating different organs. But interestingly, we saw this genetic trajectory that seems to code the visceral organs of these neuron targets along the body's rostral-caudal axis. So when we look at the animal or human, right, so organs are kind of a line along this rostral-caudal axis. And when we look at this trajectory, it seems like neurons located on one side of this trajectory tends to project to the organs close to the throat, versus on the other side of the trajectory, neurons tends to project to the lower side of the body, including the duodenum and colon. And when we plot the normalized organ positions along this rostral-caudal axis against the neuron locations on this trajectory, you can see that there's a pretty good linear relationship here, right? And remember that each organ is not a single point; it spans a fair amount of distance within the body. So if you plot the bottom bar shows the beginning and the end of the organ along this body's rostral-caudal axis, and then we plot the distribution of these neurons along this organ trajectory. You can see that they match very well, suggesting that there is a genetic trajectory that represents where the neurons are projecting along to the organs along this rostral-caudal axis. So in the following study, we noticed that we identified a second trajectory that seems to be orthogonal to the organ trajectory. And so in order to know if the second trajectory also codes essential information, we identified the genes that differentially express along this trajectory. So we see some genes are highly expressed on one side of the trajectory, some genes are highly expressed on the other side, and some genes are kind of expressed in the middle. And then we find corresponding pre-animals, and we use a genetic tool, use AAV virus encoding T tomato to label these neurons and really see what they look like within the body. So in this case, we inject the AAVs into the nodal ganglion where the cell bodies of these neurons are located, and then we take out the peripheral organs and we clear them, and then we do volumetric imaging, and then we look at how these neurons, genetically labeled neurons, projects in the peripheral organs. So on the right, I'm showing you, this is a whole mount view of a heart with the fluorescent labeled vagal sensory fibers. And then we can further zoom in of these images and look at their terminal structures at different locations. So take-home message here is we notice that neurons on one side of this trajectory interestingly tends to project through the inner side of the organs, versus the neurons on the other side of this trajectory tends to proceed to the surface region of the organ. So here, when plotting, how deep these neurons, these labeled vagal sensory terminals are located within the organ versus where the neurons are located along this second trajectory, using 5 different cray lines in multiple organs, you can see that there's also a good linear relationship suggesting that the second trajectory really predicts or represents where these sensory neurons will project to along the organ's surface to lumen axis. And next, we ask if these vagal sensory neurons form different organ-specific terminal structures, or they probably use a more generalized scheme for organizing their sensory endings. And to answer that, we labeled all vagal sensory neurons using VIGO2 animal, similar approach. And interestingly, we observed many morphological similar terminal structures across different organs. For example, in the surface elastic collective tissue layer, we observe this kind of plate of puncta type of endings, including the IGLEs on the stomach and similar endings, flower spray type of endings on the heart, versus in the muscular layer among different organs, we always see these kind of intramuscular array endings. For example, here showing you in the colon and heart. On the other hand, there's a group of specialized epithelial cells where, in the lung, there are neuroepithelial bodies. In the stomach and the throat, there are the taste buds, and the sensory neurons seem to form a similar type of endings around these cells. Then we want to ask if they are anatomically or morphologically similar across different organs. Do they actually share similar genetic signatures, or they're actually formed by different neurons with different genetics? So to test this, we take advantage of our projection seq data. For a particular organ, we find the neuron clusters that enrich for that particular organ. And then we identify the genes that differentially express among these different clusters. And then we use corresponding cray animals to label them, and going back to the same organ to see what type of terminal structures they form within that particular organ. Again, using heart as example, so using labeling all the sensory neurons, we know that where are different terminals located on the heart. And then there are largely 3 to 4 neuron clusters that mainly innervate the heart. Among these clusters, we identify 3 different marker genes. One is piezo2, one is DRD2, one is AGTR1A, and all of them differentially labeled one cluster. And then we use their corresponding gray lines and look at what type of terminal structures they form on the heart. You can see that the piezo2 form terminal structures are mainly observed in the surface as varicose endings, and the DRD2 mainly forms the intramuscular array endings, AGTR1A endings mainly as the flower spray endings. So based on this result, we can now put their terminal endings on this UMAP plot. So together, using many similar strategies across many different animal lines, across many different organs, now we summarize our data here that the red labels the ones project to the epithelial cells layer, and the blue ones represent the ones innervating the muscles, and the purple ones including representing the ones forming this punctal endings on the surface of the collective tissue of the organs. And now you can see that this also follows this tissue trajectory very well. So together, we think from both the anatomical and genetic point of view, we demonstrated that the sensory endings are kind of very closely related to the tissue layers these neurons innervate. So because these two trajectories are orthogonal, it means that they're independent, parallel, but coded in parallel rather than hierarchical. Together, they form a two-dimensional matrix that represents the body's internal space. Then we were wondering, how is the body signals, the stimulus modalities coded within these sensory neurons? Previously, in Steve's lab, we developed this in vivo calcium imaging, so we can look at the neuronal activity to different body challenges in hundreds of neurons simultaneously. This is a powerful tool, so and we can further use a Cray mouse line to label the neurons of interest and ask if this type of neurons respond to a particular stimulus. But from the single cell RNA-seq, you can see that many of the neuron clusters cannot be defined with only one marker. Actually, in many cases, we need multiple genes to precisely define a neuron population. To better understand, what neurons actually respond to what stimulus, a very talented graduate student, Yale in the lab, developed this tool. So we couple the spatial transcriptomics with in vivo calcium imaging. The idea is, so we use animal model like here, the GPR65 Cray with a Cray-dependent T tomato, and to label roughly 10% of the vagosensory neurons with T tomato signal, and we express GChem in all the neurons. And first, we do in vivo calcium imaging, and we gave challenges to the body and see what neurons are responding to these different challenges. And then after the imaging, we take out the ganglion, we cut them into serial sections, and we do multiplex RNA scope. In this case, we do rough 20-ish genes. So together that can tell us in very precisely, what neuron clusters this particular neuron belongs to. And then based on the expression of T tomato, we can register the cells between the RNA scope assays and the in vivo imaging to determine the molecular identity of these neuron responsive cells. So here I'm showing you one example. So how we do this type of registration. You can see this is a GChem. Green is a GChem signal. Red is a T tomato signal. This is from the in vivo calcium imaging. And then after the imaging, we do serial sections, and you can see that different colors represent different tissue sections. You can see on one section, we can almost like register all these super bright T tomato signals, which are in the imaging focal plane, versus if you look at all the T tomato signals above or below this imaging section, we can almost register every T tomato cells back between the in vivo imaging and the RNA scope assays. And based on, once we register the T tomato cells, we can go to the T tomato negative cells. And here I'm showing you a zoom in view of the entire images. You can see that the number 1, 2, 3, 4, and 5 are the registered T tomato cells, and based on their relationship with these T tomato cells, we can successfully register the T tomato negative cells, and then which allows us to very efficiently determine the molecular identities of these neurons. And to make a long story short, what is very interesting that we identified is, we saw is, it seems like the vagosensory neuron subpopulations defined by the single cell clustering have very similar response patterns across organs. For example, there's one neuron population, we call it C population, it's piezo2 positive expressed, and these neurons respond to stretch with a sustained kinetics to stretch in the lung, in the upper GIs, in the duodenum. Another example is a different neuron populations will respond to stretch in different GI locations with these transient kinetics. On the other hand, one thing is really interesting because usually the sensory neurons, their endings are specialized for detecting some specialized cues. But here we show that the sensory neuron endings in the vagus nerve seems to be uncoupled from their response patterns. We observed the neurons innervate small intestinal villi with almost indistinguishable morphologies. One neuron population will mainly respond to chemical challenges through the intestinal lumen. Another neuron population will largely respond to mechanical stretch of the intestine. Similar examples that the intramuscular array endings in the stomach and in the esophagus, they have similar morphologies, but they have different response kinetics versus the similar IGLEs on the stomach or on the duodenum. Some of the IGLEs endings are almost purely mechanosensitive versus others seem to be multimodal, respond to both mechanical and chemical cues. And the same type of neurons that respond to a similar challenges across many different organs. For example, this is the mechanosensitive neuron population with the sustained kinetics to different mechanical challenges in different organs. They actually form very different terminal structures in different organs. They project through this bifurcation of the lung. It forms the INAs in the esophagus. It forms IGLEs on the stomach. Although they have different terminal structures, they all sense the same thing from different organs, potentially using the same sensory mechanisms. So with all these data, we are able to put everything together and really ask the correlations between the sensory different parameters of these sensory neurons. And to make a long story short, so we can calculate their correlation index, and you can see clearly that consistent with what I just presented, that the sensory ending types are very similar or highly associated with the tissue layers that these neurons innervate. Visceral organs stand by itself, and the response patterns is quite well associated with the subpopulations defined by single cell clustering, demonstrating that these three different independent features of the interoceptive signals are coded in three independent coding dimensions. And this is, after this, we really think this is an exciting finding because we're always puzzled that there's so many different signals that needs to be coded within the system, which is composed by a very limited number of neurons. In the mouse, there are only 2,500 neurons per vagus nerve, and 2 vagus nerves. In total, 5,000 neurons are doing all this work. And it seems like it's organized in this parallel string. So how can that work? So now we think, because they are divided into different coding dimensions. And so for each coding dimension, it can only classify into a few number of groups, but in a combinatorial fashion, they can generate a large number of diversity, large number of different populations to enable this parallel string presentation of body signals from different organs to the brain. As a summary, I think our work demonstrated that there is a multidimensional architecture of this interoceptive system, that it has different coding dimensions for different features of the interoceptive signal in together, so they generate a large number of diversity to code these different information. And we think this is a perfect representation of this efficient sensory coding theory that the sensory neurons tends to use minimum resources to code maximum information. At the end, I want to thank all the people contributed to this work, in particular, very talented postdoc Chang in my lab, and a wonderful graduate student, Yu Yu, who together developed many different tools for this project, as well as our collaborators and the funding sources. And really, thank you for the invitation and for listening. Thanks.
Great, thanks, Ray, for a beautiful talk. Okay, now open for questions. Your summary slide respond.
Hi, Zilong. Hello.
Yeah, go ahead with your question.
Hi. Yeah, okay. So I have a question about the technical question. This is a great. Yeah.
Do I have okay.
Yeah, can you hear me?
Yeah.
Yeah. Okay.
Yeah,
thanks for this great. Yeah, thanks for this great talk. I have one technical question. So when you inject the retro virus to different organs, how much virus and how you use and how long you wait? Because this is a long way traveled from the organs to the brain. So how long did you wait for them?
The amount of viruses injected into different organs depends on which organ is being injected. Roughly, I think we injected 5 to 10 microliters without any dilution of the original titer, which is something around 10 to the 13th, into different organs. So we injected to the lungs through just one shot into through the trachea, but on the other organs, we tend to cover the as much areas as we can through multiple injections. We wait for 1 week, which did lots of preliminary studies and make sure this is enough time to infect the neurons.
Okay, 1 week is enough, you think?
Yeah, 1 week is enough. And we wait, even if we wait for longer, the number of neurons infected are not increasing. So.
Okay, thank you. Because we noticed that there's a tropism of the limitation of this virus. It cannot label all the virus. For example, some neurons can be labeled by rabies virus, cannot be labeled by retro AAV. But it depends on which region. So I'm just wondering whether you found that problem or in this project in SIG.
That is a good question. So we actually did a lot of controls to address that. So I think the retrograde AAVs from a number of organs that we tested have a similar or even higher infection efficiency compared to the traditional tracers, including CTBs and DIIs that people commonly use in the field. And also they label similar neurons or neuron populations comparing the retrograde AAVs and then the traditional DICE. And moreover, from the projection-seq results, we think we covered most, if not all, the neurons that project to different organs, except a couple of exceptions, including some neuron populations that innervate the lung and not in particular the bronchi are not very well labeled. Is probably because the virus does not have enough access to that location. But for most of the organs, the virus is able to penetrate into different tissue layers and location, even the deepest locations of the organ, and then infect the neurons in that way. And because we are not really analyze these neurons based on the percentage of the neurons that innervate different locations, but rather we analyze lots of neurons, and we even pay attention to the slightly enriched neuron populations labeled from these organs. So that's probably another reason why we think the coverage is pretty high.
Okay, thank you. This is wonderful work.
Thanks.
Thanks. Tai Chi, do you want to question yourself, please?
Okay, hi Ray. Interesting talk. I have a naive question about if you do single cell sequencing on the muscles that innervated by the different neurons, are you expecting the highly heterogeneity that corresponding to the neuron heterogeneity?
That's an interesting question. Well, the short answer is I don't know. I think it depends on what you're asking. It's not, we do have a rationale for why these sensory neurons are different, because they're sensory neurons. They're detecting different things, and presumably they're using different sensory machinery. So this is why we originally believe they're probably different from each other. For the muscles, I don't know, because it seems like at least it seems to me that they don't really need to be different to innervate different muscles, but
No, I mean, for example, the vagus nerve from the innervated heart, some innervated the surface, some innervated the lumen. So is there any specificity between the muscle and the nerve? That's my pairing. Is there any pairing?
You mean the neurons innervate this, like the heart muscles versus the neurons innervate the gut muscles?
No. I mean the neurons that innervate a different part of the. It looks that there is a spatial difference. I mean the inside or outside. Is there any like the specificity of this pairing?
I'm trying to understand the question. Sorry, I probably didn't really understand the question well. Are you trying to ask the neurons that innervate different muscles in different locations within the same organ?
Yeah, within the same organ. There are a lot of axonal arbors of these neurons.
We don't have, certainly we don't have the resolution to tell that. We think probably yes, because of this, because we observe really this, the axis that predicts where, represents where the neurons are going along the rostral-caudal axis or from the surface to the lumen. If we actually have a finer resolution, we probably will be able to see that even within the same organ at different locations along the rostral-caudal axis, the neurons are still following the same trajectory. There are some genetic codes to code that, but since we don't have the resolution, we won't be able to comment on that.
Okay, thanks.
Sure.
Professor Xu. Okay,
Hey, Zulong, thank you.
Yourself? Yeah, go ahead.
Yeah.
Go ahead.
Wonderful talk, Ray. I have a question regarding the whole body system physiology. So you are talking about one part is interoceptive system. My question is, what's your perspective or view about the output, like neuroimmuno interaction, like brain-gut interaction, whatever? So what's your view about these outputs?
In terms of what?
In terms of the whole body system physiology.
I mean, recently there is a paper from, or last year, there's a paper from Steve and Brett Lowe's lab working on this single cell sequencing of the DMV neurons, which is part of this this efferent arm of the nerve, and they show there are different neuron populations, and also these different neuron populations have different targets or have different downstream targets among different within different organs. I think there is likely a similar thing in the motor system as well. Also because the vagus nerve can regenerate if you damage, if it's been damaged, it can regenerate. So there's probably also similar things happening there as well, like they know where to regenerate back. But on the other hand, the motor neurons, it's probably less heterogeneous in my opinion, or my prediction, than the sensory neurons, because they probably just they don't need to have this diverse sensory machineries for different signals, and it just depends on the combination of neurochemicals they use to communicate with the downstream cells. It may be less heterogeneous than the sensory system, but we don't know much. I think we'll understand more as we get more sequencing results and more functional studies there.
So I have a second question. As Zilong mentioned, you once studied the ion channel in perhaps in Bailong's laboratory in Tsinghua. So my question is, how do you think the ion channels may play some coding roles in this interoceptive action or function?
Oh, certainly, it's one of the big players, in particular defining the sensory machineries of these neurons. For example, piezo2, the one population I showed, the C population, is expressed piezo2, and it's probably using piezo2, and it's actually been demonstrated in a number of work that using piezo2 to sense the stretch from the heart, from the lung, and it's also sensing stomach stretch, so piezo2 is probably also involved in that process. On the other hand, in addition to the ion channels, there's probably, our previous work has classified the neurons based on their expression of GPCRs, which is also like another major components that mediates this cell-cell interaction or signal interactions between the neurons and upstream cells.
Thank you.
Thanks, Taylor. So there are a few questions on the chat board, right? Can you see that? So could you quickly go through the questions from Ling? Did you see any observation with temperature changes on the simulation? I guess you've done it before.
Yeah. That's a great question. We never tried the temperature, so don't know.
Okay. Yeah, from Wang Zhuang, as I guess some technical details how to define the precise population is the one you saw with stimulus after do the RNA scope, right? How do you first align exactly the same population neurons?
So we use a number of RNA scope probes, like 20, I think it's 21 RNA scope probes here to define the 12 cell population, neuron populations we identify using a single cell RNA-seq. We have done a lot of control experiments to make sure that these 21 probes can faithfully label the neuron populations identified by single cell RNA-seq, and then it's once we know which cell respond to what stimulus, and then we can correlate with its RNA scope codes, so we know for this particular responsive neuron, what is RNA scope codes, right? And then when we have a number of neurons that respond to the same stimulus, we can look at the pattern of like what are the specific codes that highly enrich for these responsive neurons, and that's how we determine which neuron population respond to what. Right. And yeah.
Okay. The last question is: did you working on with some frozen slides by such a beautiful neural ending scanning?
It's not for the anatomical studies. It's not slides. It's a whole organ clearing and whole organ imaging. So we yeah, as I showed for the heart, that's a whole heart clear, right? And then we either use a light sheet microscope or we use a confocal microscope image at a higher resolution. I guess it's just the clearing protocols are really good, really easy to use and really nicely developed. So really thankful.
Great. Yeah, thanks. Yes. Right. That's why it's so beautiful. All right, great, thanks. All right, so I would like to thank all the really Mark for great talks and for all the insightful questions. Hi, Aaron. Thanks everybody. So for guessing today. Thank you. Thank you all next week. Bye.
Bye.
Thanks, Ray.
Bye, everyone. Good to see you, Ray. Great talk. Bye. See you.